Welcome to the Deep Dive. Today we're cracking open something pretty fundamental in cell biology, something that really changes the textbook picture.
Yeah, it's about how chromosomes get properly sorted. We're talking sister chromatid cohesion.
And the really counterintuitive finding here is this. The actual physical job of setting up that cohesion.
Yeah.
It's not done by the accessory factors we always thought were critical.
Nope. It seems to be down to the core DNA replication machinery itself. Specifically, DNA polymerases alpha and epsilon.
That's a huge shift.
It really is. And this study, it finally gives us this direct look, this visualization of the replisome, that big replication machine, actually threading itself through the cohesin ring, like threading a needle almost.
Okay, let's back up just a sec. Remind us, what's the core job of sister chromatid cohesion?
Right. So after DNA gets copied, you have two identical sister chromatids. Cohesion is basically the glue holding them together until the cell is ready to divide. In mitosis, it's absolutely essential for making sure each new cell gets one correct copy of the genome.
And cohesin is the molecule doing the gluing.
Exactly. Think of cohesin as this large protein ring, like a molecular handcuff, maybe about 35 nanometers across. Early in the cell cycle, it gets loaded onto just one DNA strand.
Right.
The big puzzle has always been, during DNA replication, S phase, how does the replication machinery, the replisome, which is itself pretty bulky, maybe 25 nanometers, get through that ring and manage to trap the newly made DNA strand inside too?
Yeah.
So both sister strands end up inside the ring.
Yeah, because the old models needed all these extra helper proteins, right? Establishment factors like ECO1, CTF4.
Correct. They were thought to orchestrate this whole complex process. But this paper asked, well, what if it's simpler? What if it's just about the physical interaction, the mechanics?
So they started with the engine of replication, the CMG helicase.
Yeah, the CMG complex unwinds the DNA. It's smaller than the full replisome, around 19 nanometers, so theoretically it should fit inside cohesin.
But there was a problem.
Well, yeah, in early tests, the cohesin ring would just slide along the DNA ahead of the CMG. It just got pushed away. So the clever trick here in figure 3 was that they used an antibody to basically tether the cohesin ring, immobilize it, stop it from sliding.
Like holding it in place, and what happened then?
Then passage became the most common outcome. About 40% of the time, when the CMG hit the stuck cohesin ring, it went through.
It just pushed through.
Well, not effortlessly. They saw the CMG stall for a bit, then make these visible jumps forward. It suggests force is building up. And they calculated this force was less than 20 piconewtons. Importantly, this is enough to maybe stretch or unfold the cohesin wing a bit, but not break it. And later figures confirmed the DNA stayed trapped, so the ring remained intact.
Okay, wait. That brings up a thinking point for you listening. If the core helicase, the CMG, can generate enough force to kind of stretch and pop through the ring, what happens when the entire replisome, which is much bigger and bulkier, hits that ring? Does the extra size block it or maybe help it?
It's a great question. You'd intuitively think bigger means harder to get through. Right. But here's the fascinating part, shown in figure 5. They built the full eukaryotic replisome and watched it encounter cohesin. And yes, passage was less frequent than with just CMG. It happened in about 18% of encounters.
Okay, so less frequent, but it still happened.
It still happened. And crucially, this wasn't just about getting through. In the majority of those passage events, 31 out of 42 they recorded, it resulted in stable sister chromatid cohesion. They could literally see the new DNA strand being held together with the original template strand by the cohesin ring after the replisome went through.
Wow. So that's the direct physical proof. The replisome going through is how cohesion gets established.
That's the core finding. The transit itself does the job.
Which leads to the million-dollar question in figure 6. What part of that big replisome is essential for making this happen? Was it those classic establishment factors, after all, maybe working with the main machine?
So they tested that. They built replisomes missing some of those factors, like CHL1, and it didn't really change the passage frequency much in their clean in vitro system.
Huh. So not the usual suspects.
Not primarily, no. The real shocker came when they started removing core DNA polymerases.
The enzymes that actually synthesize the new DNA.
Exactly. And get this. Replisomes completely lacking DNA polymerase alpha, they never pass through the cohesin ring. 0 times out of 41 tries.
0. Seriously.
0. Total block. And they also found that a part of polymerase epsilon, specifically its non-catalytic C-terminal tail, was also really important for helping the passage happen.
So pol alpha is essential, and pol epsilon helps. These core replication enzymes are acting like like molecular crowbars or guides to get the machine through the ring.
That seems to be the implication. They are the key molecular determinants for this physical threading. It's not just about synthesizing DNA. Their structure is physically involved in navigating the cohesin barrier.
This is really paradigm shifting. It provides that missing kinetic link, showing cohesion isn't some separate add-on process, but like a direct geometric consequence of how replication happens.
Precisely. It elevates this replisome transit hypothesis right to the forefront. And it means we might need to rethink things like cohesinopathies diseases linked to cohesin defects, or even how replication stress affects chromosome stability. Maybe it's about altering the probability of this passage.
Yeah, the textbooks definitely need an update, prioritizing the polymerases in this role.
Absolutely.
Okay, let's do a quick recap of the key takeaways for everyone listening. First, the replisome physically shoves its way through the cohesin ring to set up sister chromatid cohesion.
Second, this passage isn't easy. It involves force, enough to stretch the ring temporarily, maybe up to 20 piconewtons, but the ring stays intact.
And third, the absolute non-negotiable parts of the replisome needed for this threading are the core DNA polymerases, specifically Pol alpha and also Pol epsilon's tail.
Right, which leaves us with a really interesting thought to ponder. If these polymerases are key for passage on naked DNA in vitro, what about those classic establishment factors identified through genetic studies? They didn't seem essential here.
So what's their job then?
Well, maybe the cell has parallel pathways. Perhaps this polymerase-driven physical passage is the default on clean DNA, but when the replisome hits obstacles like tightly packed chromatin or other complex DNA structures in vivo, maybe that's when you need those accessory factors to come in and help manage the process.
So context matters. The clean lab setup versus the messy reality inside the cell nucleus.
Exactly. It suggests there's still more complexity to uncover about how the cell ensures robust cohesion under all conditions. That's definitely the next frontier.
Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has three main goals. 1, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. 2, to build a supportive community where we can exchange the most valuable experience-based research advice. 3, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.